Thank you for the introduction, and hello everyone. I would like first to say thank you to every one of you for attending today's webinar entitled "The Human Ex Vivo Heart Model for Drug Discovery." This slide, I show you the items that I'm going to cover in my webinars today. First, I will cover or explain the human tissue procurement we do at Anabios and how we did develop the ex vivo human cardiac models. Then I will explain how these models can be used to assess the cardiotoxicity potential of novel drugs and how they can be used to help and support development of novel cardiac therapies. And then quickly, I will show you some new models that we have recently developed and finish my webinar with the take-home message. Anabios is a unique company dedicated to human translational sciences and studying the effect of novel therapies in human tissues. To this aim, we work with a human organ procurement organization in the United States only to recover ethically consented human organs. To do this, we have well-trained surgical teams. Also, we have developed a proprietary solution for recovery and shipment of organs. The solution ensures high viability of the recovered organs. Of course, we have standardized our method, and of course, each organ comes with an extensive annotation. One of the organs we recover is the human heart, and the recovery rate we have allows us to support discovery projects during nomination of drugs phase or during early drug discovery. As you can see on this slide, for nomination of drugs, we have tissue-based models to assess the potential of drugs, to affect electrical activity and electrophysiological function of the heart. We also have the models, to assess the potential of drugs to modulate contractility of the heart, also to modulate the blood pressure, and also to modulate the conduction of the heart. As I said, these models can be used for nomination of drugs and during later preclinical development. To support optimization of drug during early drug discovery, we have cell-based models, and this is where we isolate cardiomyocytes from the human heart to conduct contractility experiment, and also we can run studies to assess the potential of drugs to affect calcium dynamics. In addition to the normal hearts, we also can recover diseased hearts. And this is where, again, our main activity and work is focused on nomination of drugs where we can run, again, the contractility experiment. As you can see from this slide, we can recover hearts from donors affected by heart failure. And here, we can get HFrEF and heart failure with a preserved ejection fraction. Also, we can get hearts from donors affected by hypertrophic cardiomyopathy and coronary artery disease. And also we can recover hearts from donors affected by atrial fibrillation if a company is working to find novel treatment or novel antiarrhythmic to treat the patient with atrial fibrillation. As you can see on this slide, the model we developed are very well validated, and the data were published, have been published in peer-reviewed journals. As you can see, we have done a lot of validation. We have tested a lot of reference compound, and all this data are published and available for you if you would like to look at the details or the full details and the full data set in these publications. In addition to publishing our validation data, we also collaborate with regulators. And as you can see on these slides, As you can see, we have 4 ongoing collaboration and project with the FDA. So we generate data for the agency to better understand, the human biology or the biology of the human heart when it comes to the electrophysiological activity of the heart, and when it comes to the contractility, the mechanical activity of the heart, both at the tissue levels and also at the cellular level. I will now show you examples on how the models can be used to assess the cardiotoxic potential of novel drugs. The first model I will present to you is human ventricular action potential models. These models can be used to assess the potential of novel drugs to prolong the QT interval, and also it can be used to help assess or bridge the gaps when it comes to the E14S7B Q&A document. And again, if you and any of your colleagues are attending the annual meeting of the Safety Pharmacology Society meeting in September next month in Brussels, we will present data that we generated as part of the collaboration with the FDA when it comes to the action potential ventricular model. As you can see on this slide, the slide explains to you, how we recover the heart, how we process and dissect the ventricular trabecula, and how we stabilize, the trabecula under the experimental condition needed to record action potential. And as you can see, Anabios has multiple rigs, for conducting action potential measurement, where we can run multiple experiments at the same time, where also we can test multiple compound and different concentration at the same time. As you can see from the slides, we published data with this model with 18 reference drugs. 11 of them are torsadogenic, 7 of them are non-torsadogenics, and 13 of them were tested blind. Of course, we have unpublished data with additional 15 reference drugs. As you can see from the data we published, we have a low inter and intra-donor variability is approximately around 7%, and the sample size needs to test the effect of a drug in this model is 4 tissues. Here is some data to show you that when we record the action potential duration from this model, we have good stability over time. So if you assess the effect of a drug and you see an effect, it will be compound related and not due to unstable recording over time. As you can see, also, we have determined the activity threshold for different parameters of the action potential, especially when it comes to the duration of the action potential at different levels of repolarization. And also we did determine, the activity threshold when it comes to proarrhythmia markers like triangulation and short-term variability of the action potential duration. And also, I will show you today data with the 3 critical compounds that they are important when it comes to the E14S7B Q&A document. And these 3 compounds are moxifloxacin. As you know, moxifloxacin is a compound used in soro QT studies in clinical trials. We will show data with dofetilide and ondansetron that also known, to prolong the QT interval and be associated with torsadogenic potential. As you can see on these slides, we are presenting, the change in APD90 duration of the action potential relative to concentration and also relative to multiple of the therapeutic free Cmax. As you can see, when looking at the dofetilide, moxifloxacin, and ondansetron data, you can see that the 3 compounds showed the potential to increase the action potential duration, and this is how we confirm in this model their ability to cause the QT prolongation in patient. In addition to the action potential data, we can also look at their potential to cause manifestation of proarrhythmia markers. And here I'm showing you data with triangulation. Again, you can see on the y-axis, this is a percentage change in triangulation versus the concentration we tested for the 3 different drugs. And you can see we compare the proarrhythmic manifestation of the proarrhythmic markers to the therapeutic free Cmax and to the HERG IC50 when that were generated with these 3 compounds. And again, you can see that dofetilide, ondansetron, and moxifloxacin, in addition to prolonging the action potential duration, were able to cause a manifestation of proarrhythmia markers. And again, this is the way or the approach we use to determine if a compound will prolong QT and if the compound will be associated with the proarrhythmic potential. And of course, all this data are generated at the preclinical stage, well in advance of a clinical trial. And the hope here is that this data will play a critical role in helping you to integrate any additional cardiovascular data you have from other models and to make the decision of taking forward a compound or stopping a compound for cardiotoxicity potential. Now I will show you how we use the isolated ventricular myocyte to assess the proarrhythmic and inotropic potential of novel drugs. As you can see here on this slide, we are showing representative images of ventricular myocyte. As you can see, this myocyte have the rod-shaped morphology. They have a clear striations that is representative of the contractile machinery in this myocyte. And most importantly, this myocyte, they don't beat spontaneously. You need to apply an electrical field stimulation for this myocyte to contract and relax. And this is how this myocyte do work in your hearts and in my hearts. They need the electrical stimulation to be propagated, from the pacemaker all the way through the atria, the conduction system, and then the signal will reach, this ventricular myocyte, and they will contract. So this myocyte, as I said, they have different morphology, different structures, and different calcium dynamics when you compare them to the human iPS-derived cardiomyocyte. And most importantly, they don't beat spontaneously as they are mature myocyte isolated from adult human heart. At Anabios, we developed the MyoBlazer, which is a novel platform that allows you to preserve the normal physiology and pharmacology of the cells. And most importantly, it allows you to have a continuous measurement of contractility from multiple cells in parallel. And you can apply, you can determine the concentration dependency of your compound. You can do 1 concentration up to 4 concentration. You have the same exposure time to a drug and to a concentration. And of course, a big advantage that you have a real-time tracking of the drug effect throughout the experiments. We have automated acquisition and automated analysis. This helps us, to generate data, analyze data, and report data very quickly to you. If you would like to read more about the MyoBlazer, we did published recently a paper in Jove where we did present the characteristic of this new platform. On this slide, we show you, the different parameters that we can derive from the contractility transient when we record contractility from these cells using the MyoBlazer. You can see we derive multiple parameters. It gives you an idea about the effect of drug on the systolic phase of the contractility transient, but also it provides you information on the relaxation phase of contractility. And all these parameters can be used to characterize compound and also to get an idea about a potential mechanism of action when it comes to your compound. Here is some data from the MyoBlazer, and as you can see here, this is the time control to show us that the contractility transient we record from the cells are stable over time. And here, we did mimic an experiment or a drug experiment, with vehicle 0.1% DMSO, and each application of the vehicle was for 5 minutes. So you can see we start the experiment with 5 minutes under baseline recording, and then we mimic an experiment with a drug at 4 concentration, and you can see we were able to record from multiple cells, and you can see that we have a good stability over time. So this means that if we test a drug in the MyoBlazer system and we see an effect, it will be highly potentially that it's a drug related and not an effect due to instability of the contractility transient we are recording using this new platform. This is an example with a compound that decrease contractility. So it is the same design. We are recording here from 5 cells at the same time. We have good baseline stability. And then we did test the compound at 4 ascending concentration. You can see first concentration of the compound has no effect. And then second concentration, we start seeing the effect on contractility. Around the third concentration, we can determine the IC50. At the 4th concentration, you can see we have the maximum effect on contractility. And after this, we have the ability to wash out the compound and determine if the compound effect is reversible or not. In addition to contractility, we can check the effect of compound on calcium dynamics in myocyte. When we do this experiment, they are run separately from the contractility. So for now, the MyoBlazer is a system that allows you to record contractility, and we hope in the future that we will bring to the MyoBlazer the calcium imaging capability too. But for now, the two models are run separately. As you can see from this video, we are recording from 26 myocyte at the same time. And again, we measure calcium dynamics here and effect of drugs on calcium dynamics. And similar to contractility, we can derive multiple parameters, so we can assess the effect of the drug on the peak height of the calcium transient, on the peak width at 50% peak height, and also we can assess the effect of compound on diastolic calcium. Our recommendation is to test first the effect of your compound on contractility. If you see an effect, the calcium imaging can be used as a follow-up study to determine if the effect of your compound is calcium mediated or independent of calcium. Now I will explain to you how the contractility measurement can be used to derive in the same experiment the potential of a drug to affect the contractility and to cause arrhythmia. So basically, the torsade de pointe and the proarrhythmia is not a pure electrical issue or problem or toxicity in the heart. There was a lot of clinical data showing that long QT syndrome and torsade de pointe proarrhythmia is an electrical problem and a mechanical contractility problem when it comes to the heart. And this tight relationship between ECG and contractility mechanical activity of the heart, we can see it at the level of single cardiomyocyte. So this is the relationship between the action potential and the contractile machinery in the single myocyte. So previously, companies to assess the potential of drugs to cause proarrhythmia and effect on contractility, they were running 2 separate models where in one model you record action potential, in the second model you record contractility. So what we are proposing here, is to replace the 2 models with one model. As you can see on the contractility transient here, the blue part of the contractility transient, so this is the sarcomere shortening. We will use this part of the signal to derive inotropic effect of drugs, but also we will be looking at the relaxation phase of the contractility, where we will record the after contraction events as a proarrhythmia markers, and the relationship between early after depolarization and after contraction is well established. And basically the after contraction here mimics the premature ventricular contractility that usually leads to trigger early after depolarization. Here is an example how we can differentiate dofetilide, a proarrhythmic compound, from verapamil, which is a safe compound or non-proarrhythmic. As you can see here with dofetilide, when we apply dofetilide at the 20 nanomole, we can see the incidence of after contraction highlighted here in red. And when you compare dofetilide to verapamil, you can see that verapamil showed no ability to cause after contraction events. However, we were able to see the effect of verapamil on contractility because it is a calcium channel inhibitor. So this is how we use the models and the after contraction and sarcomere shortening to differentiate the proarrhythmic drugs from non-proarrhythmic drugs, and to differentiate the compounds with effect on contractility from compound with no effect on contractility. Here is some of the validation studies that we did publish, and you can see here in this table we show you data with 23 proarrhythmic drugs, and you can see testing these drugs in our model and analyzing the data at tenfold therapeutic exposure using the after contraction as a marker. You can see we were able to detect most of this compound, and showing that the model has high ability to predict positive controls. When you compare our data to human IPS-derived cardiomyocyte data published by Amgen and also published by different initiatives and also by FDA, you can see that the human IPS cardiomyocyte had a higher potential to provide false positives. Here are the data, with 10 non-torsodogenic compounds, and you can see again using the after contraction as a marker, we were able to predict the true negative potential of these drugs, and again the human IPS myocyte were associated with false positive predictivity. Again, as I said, from the same data, we were able to derive the contractility effect of this 33 drugs. As you can see here, we were able to do dose response curve and derive IC50 data, and also derive safety margin for all these compounds. This is another example, during the COVID-19 pandemic, we did a joint study with Novartis to assess the cardiotoxic potential of hydroxychloroquine. As you know, hydroxychloroquine was proposed as a compound to treat COVID-19. As you can see here from the data, you can see at therapeutic exposure, hydroxychloroquine showed no potential to be proarrhythmic. When you go 30-fold above therapeutic exposure, so this is when you start inhibiting the HERG channel and the CAV1-2 channels, you can start seeing a proarrhythmic potential and effect on contractility. When we compare the hydroxychloroquine to chloroquine, you can see with the chloroquine we saw proarrhythmic potential at therapeutic exposures, and of course we saw a lot of arrhythmic and proarrhythmic events when we went also above therapeutic exposure. And this is how we were able to differentiate the safety of hydroxychloroquine from chloroquine at therapeutic exposure. We did a lot of risk factor when we did this study, like hypokalemia, fever, so that they are risk factors when it comes to COVID-19 patients. All this data were published in Toxicological Science journal. In addition to hydroxychloroquine, this is another example for AZD7762. This compound was developed by AstraZeneca as an anti-cancer agent. You can see left-hand side of the slide show you that all the data that were generated using animal models and using cell line or using dog myocyte. As you can see, the model showed a potential to cause cardiotoxicity, but the data indicated a safety margin of around 15%. However, when AstraZeneca took this compound to clinical trial, you can see there was a very significant effect on contractility in patient, and they were forced, to stop the clinical trial. When we tested AZD7762 in the human primary cardiomyocyte contractility models, you can see that we were able to predict the risk, and our data were able to determine that the compound has less than 1-fold safety margin to the risk versus a 15-fold margin to the data generated using dog myocyte. Another example is AZD9291. So again, this is a cancer compound developed by AstraZeneca too. And you can see we tested this compound in our models. You can see at therapeutic exposure, the compound showed no ability to affect contractility and also showed no ability to cause proarrhythmia. Of course, when you increase the concentration well above therapeutic exposure and exceeding the Herd IC50, this is where you start, seeing some proarrhythmic potential and some also ability to decrease contractility. Also, the contractility model can be used to better predict compounds that may cause the prolongation of the QRS complex. And again, this is a study that was published by AstraZeneca, where the conclusion of the study is that the QRS prolongation occurs on average at a free plasma level 15-fold below the IC50 at NaV1.5. And as you can see, from this data, we tested the same compounds that were reported in this NaV1.5 study. And you can see, using the contractility model and looking at the risk window for QRS risk and also looking at the safety margin, you can see that the human primary contractility, myocyte contractility model showed a better ability to predict the QRS risk. Now I will show you how the model can be used to develop novel cardiac therapies. The first example that I will show you, it's a drug X that has been developed to treat atrial fibrillation. As you know, atrial fibrillation, you need to have a compound that shows atrial selectivity, but at the same time, the compound should not affect the ventricle function and should not be associated with proarrhythmic potential. As you can see on this slide, we tested the effect of the compound in myocyte isolated from AFib hearts. As you can see, we did show that the compound prolonged the action potential in atrial myocyte, but also we did demonstrate the compound tested in atrial trabecula isolated from donors affected by atrial fibrillation. You can see already at baseline recording, we were able to see arrhythmic events like after contraction and triggered contraction. And then when we tested the compound at 0.1 micromole, which is very close to the predicted free Cmax, and the effect, the IC50 for the effect to inhibit an ion channel, you can see that we did block the arrhythmic events, and we did recover the normal activity of the atrial contraction. And of course, we did determine the dose response curve for this compound. So this data are important, and they are like a proof of concept data at the preclinical stage to demonstrate that the drug X has the potential to be an anti-AFib drugs. Next, we wanted to demonstrate that the compound is safe and has no effect on the ventricle. And again, you can see here we tested the effect of the compound on atrial myocyte, on action potential from atrial myocyte in normal heart. And also we did assess its effect on ventricular action potential. You can see the compound did not affect the action potential duration. Also, we tested the compound effect on ventricular myocyte contractility and also on force of contraction measured from ventricular tissue. Again, the compound had no effect to modulate contractility and did not show, again, a potential to be proarrhythmic. So this is how we can confirm that the compound is atrial selective and has no potential to cause cardiotoxicity when it comes to the ventricular function. Again, this is another slide to demonstrate that this contractility model can be used to assess potential of drugs to be anti-AFib. Again, this is data from two different AFib atrial trabecula. As you can see, we can see arrhythmic events after contraction events, and we can see premature atrial contraction when we tested a late sodium channel inhibitors, GS967. You can see that we were able to block arrhythmic events, and we were able to recover the normal function of the atria in this AFib hearts. Also, the ventricular myocyte we isolate can be used to develop novel therapies that are based on modulation of heart contractility and relaxation. As you can see on this slide, there is effort, to develop treatment for heart failure with reduced or preserved ejection fraction, and also for obstructive hypertrophic cardiomyopathy and antiarrhythmic therapy. And the next few slides, I will show you how the ventricular myocyte can be used to support treatment of diseased human hearts. This is data showing data with myosin modulators. As you know, myosin modulators, when it comes to omecamtiv or when it comes to mavacamtiv, have been or are being developed, to treat heart failure with reduced ejection fraction. And when it comes to mavacamtiv, it has been recently approved by FDA for treating obstructive hypertrophic cardiomyopathy. So here, we are using again the myoblazer, and you see here we did test different myosin inhibitors, and you can see we did test mavacamten as one of them. You can see we have good reproducibility from within the same heart and also between hearts. And you can see, compound like mavacamten, compound like BTS and blebistatin, they show dose-dependent decrease in contractility. Also, we did test analogs of blebistatin, and we can see biphasic effect of blebistatin analogs. So the conclusion or the message from this slide is that the myoblazer and the myocyte can be used to support early chemistry and also to provide the SAR data to early drug discovery programs. Here are some data to show also in addition to inhibiting the myosin and showing decrease in contractility, we can also test myosin stimulators, and here omecamtiv is a good example. As I said, omecamtiv is being developed for the treatment of reduced heart failure with a reduced ejection fraction. And you can see here, omecamtiv tested at 10-fold therapeutic exposure in different hearts. You can see that we were able, to see the increase in contractility caused by omecamtiv. And again, this data demonstrates the ability of the model, to predict clinical effect when it comes to omecamtiv and to myosin stimulators. Again, here is some data to show that we can derive mechanistic understanding from the data. And here we are comparing the omecamtiv effect versus application of extracellular calcium on different parameters like rise time, like time to 90% relaxation, and also looking at the sarcomere length. And you can see the difference between omecamtiv and extracellular calcium. You can see extracellular calcium does not cause any change in these different parameters, where omecamtiv was able to cause changes in this parameter. So this is how we can differentiate and get some mechanistic information from the data that we do generate. Again, here we did follow up testing of myosin modulators on calcium imaging. As you know, this myosin modulators are being developed to treat heart failure because their effect is not calcium dependent, and therefore using this compound you avoid the potential to cause arrhythmia when you treat a patient with heart failure. And you can see here we are showing data with mavacamten at 300 nanomole, which is the therapeutic exposure, and the IC50. For the compound, you can see the compound showed no potential to modulate intracellular calcium, and our positive control, verapamil, was able to decrease intracellular calcium. So when we tested the omecamtiv, mavacamten, and the other different myosin modulators, you can see they showed no ability to modulate intracellular calcium. And again, using the contractility and this intracellular calcium data, we can provide mechanistic understanding in addition to analyzing and determining the effect of the compound on contractility. Also, in addition to this, we, as I said, we recover diseased heart, and here, I'm showing you example from left ventricular trabecular contractility measurement using tissue isolated from donors affected by preserved ejection fraction heart failure, and you can see here, we did look at baseline parameters when we do this recording. You can see here we did look at the time fall. Basically, this is the time needed for the contractility to fall from peak contraction to full relaxation. We did compare this the time needed to reach the peak of contractility. Also, we did look at the time needed to 50% relaxation and time for 90% relaxation, and we did compare normal heart to HFpEF heart, and you can see we were able to detect the impaired diastole that did translate here well, from the donor heart to the tissue data that we generate after receiving the heart in our lab here in San Diego. Also, this is some data showing you the utility of using the diseased human hearts data. And here, we did compare the effect of mavacamten on normal contraction versus HFpEF contraction versus HFrEF contraction. And you can see from the data that mavacamten showed a stronger ability to decrease contractility in HFpEF relative or compared to HFrEF. So this means that mavacamten will have better potential, to treat patient and control contractility in HFpEF patient compared to HFrEF patient. And you can see here, it's not a surprise that BMS now are conducting a phase 2A clinical trials with mavacamten to assess its potential to treat patient. And this clinical trial and the data we generated strongly suggest that mavacamten can be a good treatment for HFpEF patient. In addition to HFpEF, also we can use the models, the tissue contractility model, to support novel treatment for heart failure with reduced ejection fraction. And this is some data that we did publish with Arena Pharmaceuticals. Arena Pharmaceuticals have developed a beta-3 antagonist for HFrEF treatment. We did work with them for over 2 to 3 years, generating data for their beta-3 antagonist, and also we did compare their compound to many reference clinical compounds. And the idea here is that in HFrEF patient, you have overexpression of beta-3 agonist, and you can see here on the slide, we did provide tissue sample for them to confirm the overexpression of the beta-3 receptors. And the idea here, when you block the beta-3 receptors, basically you will block nitric oxide generation and cyclic GMP production. And by doing this, you will relieve the impaired contractility. And this is how you will treat the patient. And you can see from the data here presented at the top right-hand side of the slide that when treating the tissue, the HFrEF tissue with this beta-3 antagonist in the presence of norepinephrine, you can see that we were able to increase the contractility in HFrEF patient versus normal donor hearts. And this is how we confirmed or provided proof of concept data to Arena Pharmaceutical that the compound has the potential to affect contractility in HFrEF patient, and now the compound is in phase 2 clinical trials. And now I will show you some new capabilities and new models that we did recently developed. One of them is the possibility to ship myocytes. So all the data I showed you in previous slides were generated using myocyte isolated here in our lab in San Diego. However, we wanted to be able to provide the myocyte to pharma companies, to academia, and to CROs, so people can have this myocyte in their labs, be able, to generate their own data, and also to execute any experimental designs they would like to run. And first, to start doing this, as you can see here, we published the data recently. We had a collaboration with Amgen that is based in Thousand Oaks. And as you can see, we demonstrated that we can transport the myocyte after isolation for 3 hours transportation. And as you can see, as you can find in the paper, the Amgen was able, to record the contractility and calcium dynamics simultaneously from this myocyte, and showed that after 3 to 4 hours transportation, the cells were viable and can be used, to do pharmacology and testing of drugs. And of course, after this validation, we wanted to be able to ship myocyte to other places in the United States, but also to be able to ship the myocyte to worldwide. So what we did first, we did ship myocyte, and test the shipment of myocyte from San Diego to different airports in the United States. And this is an example of the myocyte that they flew from San Diego to Florida, and then from Florida back to San Diego. And when we tested them in our lab, as you can see here, we are using the MyoBlazer again. We are recording from multiple myocytes at the same time. After air shipment, the cells kept their morphology, they kept their ability to contract and relax when we apply electrical stimulation. As you can see on this slide, we demonstrated that the baseline contractility was not different between shipped and non-shipped myocyte. And also we were able, to see a good response to isoproterenol, a non-selective beta adrenergic agonist. And also we were able to see a good response to milrinone, a PDE3 inhibitors. And again, there was no difference between shipped and non-shipped myocyte. As I said, now we are collaborating with multiple pharma company, multiple instrumentation companies, and multiple academia lab, where we are sending, our myocyte so they can be tested and validated following shipment. This is an example from myocytes that we did send from San Diego to Physiosteam, which is a CRO in France. And you can see from this slide that the cells arrived viable to France, after a few days of shipment. They were able to record action potential, and they were able also to see the effect of dofetilide. So here, as you can see at the bottom right side of the slide, action potential in black is baseline control action potential, and when we did apply dofetilide, we were able to see the action potential prolongation. So this is some preliminary data, showing that the cells stay viable, stay functional, and you stay responsive, to different drugs. In addition, to be able to ship myocyte, we also developed a models where we can keep the myocyte in culture for 24 hours. As you may know, when you put the primary myocyte in culture, they usually de-differentiate and revert to embryonic fetal phenotype. So we managed to develop a new protocol where we can keep the cells for 24 hours. And you can see on this slide, the cells, kept their rod-shaped morphology. There was no change in the structure. They were still able to contract and relax. And this is some data where we showed, that the compound were responsive to ISO and also were responsive to ranolazine and verapamil. And we were able also to detect a proarrhythmic event, when we did the test dofetilide and beta-adrenergic agonist like ISO. We have used, this protocol to support multiple companies when it comes, to cardiotoxicity assessment, but also when it comes to test peptides, antibodies, and ASO for heart failure treatment. I would like to finish my presentation with the take-home message. The take-home message here is that human ex vivo models are now available at the preclinical stage of drug discovery to study the drug effect and of novel therapies. And hopefully the data here will play an important role to identify effective and safe therapies. Certainly the data will help you to determine if your drug work in a human. It will help you to identify cardiotoxicity risk of novel drugs. It will help you to identify patient population for specific treatment and specific drugs. And of course, it will help you to get accurate prediction of dosing in patient and to provide you important information on your safety margin when you go and dose your compound in patient and in clinical trials. Thank you very much for your attention, and I will be very happy to take your questions.